Hi there, I am Patricia Ahl. I work in the Translational Immunology lab at A-STAR in Singapore, and I'll be sharing our work on the development of MetFlow, which is a single-cell protein-level analysis that reveals metabolic remodeling in immune cell populations. This is a novel single-cell metabolic profiling technology using flow cytometry. The metabolism is a process that provides energy and building blocks that are central to sustaining life. And there are four main macromolecules which really maintain cellular homeostasis, and they can be used as fuel sources by regulating their anabolism and catabolism. So this is really the foundation of cellular function. And in homeostasis, but also in the context of disease, understanding metabolism on a single cell level is critical, especially in heterogeneous environments, such as the tumor microenvironment, where multiple different cell types, such as immune cells, stromal cells, and tumor cells, are in dynamic interaction with each other. And so we applied MetFlow to unravel immune cell metabolic population profiles. There are many methods that currently exist, and these are all good in their own right. So extracellular flux analysis is a real-time respiration measurement for oxygen consumption as well as extracellular acidification rate. However, this is a bulk level analysis. Fluorescent metabolic dyes are great for live uptake of glucose, for example, but these are very difficult to multiplex. And lastly, of course, single-cell RNA sequencing is a great way to look at gene expression analysis, but this is not always correlated to protein-level analysis, and it does not include post-transcriptional and post-translational modifications, which are significant in the regulation of cellular function. So we developed MetFlow to overcome these challenges, and this is a single-cell profiling technology. It is high throughput at the protein level. We are specifically measuring the capacity for flux of rate-limiting enzymes and critical proteins, which can then be correlated to the functional and phenotypic analysis on a single cell level. We built this technology by focusing on 10 metabolic proteins, which we then combined with 18 phenotypic markers, and these are the pathways that are highlighted here, which we can then measure using this MetFlow technology. So firstly, we showed that if you just look at the expression of the 10 metabolic proteins as well as the 18 immunophenotypic markers, we have a nice separation of cells distinctly by their different cell types. And interestingly, when you then compare each cell type to the specific metabolic proteins, there really is a positive and negative correlation of cell types to metabolic proteins, which indicates a differential metabolic state. More importantly, when you use only the expression levels of 10 metabolic proteins and ignore the immunophenotypic markers, these expression profiles are really strong and can identify immune cell populations based on their metabolic expression alone. And when we compare these 10 metabolic proteins to 500 metabolic genes by single-cell RNA sequencing, this is publicly available data, we actually have a comparable resolution to single-cell RNA sequencing. Of course, if you look at 10 metabolic genes alone, this is not the case. So using this technology, we then focused on T cells, and at rest, you can see that these T cells memory subsets identified by CD45 and CCR7 really have differential metabolic expression levels as well. More importantly, though, in the context of activation and glycolytic inhibition by using CD328 and 2-FDG respectively, we have a shift in the metabolic programming as well as the immune cell subsets. And we really have this distinct population of central memory cells that increases with glycolytic inhibition and activation. And this in green, and this is very distinctly different if you look at the t-SNE projection from the untreated and the activated cells. The activated cells are in orange, and they have a different metabolic profile, which is a reduced capacity through several metabolic pathways in comparison to activated cells. We then wanted to tie this together with the functional aspects of T cells. GM-CSF production is important in T cell inflammatory function, and it can be a protective role, but also detrimental when T cells produce too much GM-CSF, and it can cause tissue damage. And we measured GM-CSF by using a GM-CSF detection antibody to combine this on a single cell level with MetFlow, and we're able to see that the central memory population under glycolytic inhibition produces a very high amount of GM-CSF, and this is metabolically distinct from the other cell types. So really, MetFlow is a strong technology that correlates the metabolic state to immune cell phenotype and function. This is a single cell resolution, so it can be, we can increase the complexity. It overcomes existing challenges of technologies, and interestingly, we can use this on any different cell type and any different disease, and then hopefully this can lead to the discovery of novel axes that can be metabolically targeted if we can just take this high-dimensional analysis further. And yeah, approach me if you have any questions. Thank you very much.